All right, the last thing we want to cover in this chapter would be modifications of the cell surfaces. We're going to talk just a little bit about cell surfaces in animals and then cell surfaces in plant walls as well. But we're going to talk about two specific types of animal surface features. First thing we're going to talk about is the extracellular matrix. This is going to be found just outside of the cells, and it's going to form the junctions between some types of cells. Both of these can be used to connect to the cytoskeleton and contribute to the communication between cells and therefore tissue formation. The extracellular matrix is a meshwork of proteins and polysaccharides in close association with the cells that produce them. So this is just a little bit of what that extracellular matrix looks like in an animal cell. So here we have our collagen. Collagen is going to be a protein that's going to help resist stretching. So that's this red fiber here. It's going to help maintain elasticity inside of our skin cells and inside of our tissues. Elastin, on the other hand, is going to help provide resilience. It's going to allow our material, or collagen gives us shape and structure. Elastin gives us elasticity, the ability for our skin to recover or our tissues to recover after they've been stretched and moved. Fibronectin is actually a very interesting molecule. Fibronectin is an adhesive protein that binds to integrin. It is going to connect collagen to integrin, and integrin is going to be bound to our cytoskeleton on the inside of the cell. This connection right here between fibronectin, integrin, this adhesive protein found in the plasma membrane, is also going to be bound to the actin filaments of the cytoskeleton. So this connection that we have right here is what allows our cells to be somewhat anchored. It anchors them not only to each other, but it anchors them to the extracellular matrix as a whole. We have our interior cytoskeleton anchored to this integrin protein, bound to the fibronectin, bound to these collagen fibers on the outside. So this is what sort of anchors our cells in place, even though the plasma membrane can move very freely around those integrin proteins. The last molecule that we want to mention would be proteoglycan. Proteoglycan is a long polysaccharide, that's going to be this molecule right here, that's going to resist the compression of the extracellular matrix. Basically, it's like great big cotton candy stuffing. It's going to provide a sort of a spongy, open matrix between our cells to prevent seriously close packing. So that extracellular matrix, like I said, it's a mesh of proteins and polysaccharides. Here is going to be just another slide that's going to help summarize what all of those different molecules do, in case you didn't get a chance to write them down before. All right, and then the next thing we want to talk about is junctions between cells. So we talked about the extracellular matrix found outside of the cells, but now we're going to talk about how cells can actually be connected to each other through junctions. Because in the case of multicellular organisms, we're going to be forming a lot of different tissues. We need those cells working together to form tissues, those tissues working together to form organs, working together to form organ systems, and then we've got our organism as a whole. This can't be done unless these cells work together and communicate. So there are certain tissues in vertebrate animals that are known to have junctions between their cells that allow them to behave in a coordinated manner. And there are three types of junctions that we're going to talk about. The first is an adhesion junction. Adhesion junctions mechanically attach adjacent cells with intercellular filaments between the cells. So the intercellular filaments between the cells, they use one type of adhesion junction, would be a desmosome. A desmosome, there's going to be an internal cytoplasmic plaque that are attached to the cytoskeleton of each cell, and then they're joined by integral membrane proteins called cadhedrans. This results in a really sturdy but flexible sheet of cells. So you can kind of think of it as like buttons on a coat. These two cells are next to each other, but they're not really attached except for right here at these desmosomes, right here at that little junction. So it's kind of like buttons. They're not going to hold everything together, but they're going to hold the cells in place with these little connections. Now there are also tight junctions. Tight junctions are also adhesion junctions. We're still trying to adhere two cells to each other, but it's a tight junction. It's going to bring those cells even closer together than those desmosomes. This is going to be more like a zipper. we're going to see really, really, really tight connection. We're going to see almost no space in between those cells, no room for error. These tight junctions are going to connect the plasma membranes between adjacent cells together. This is going to produce kind of like a zipper-like fastening. And this is going to be very important for tissues that serve as barriers, like your intestines. We want to keep the digestive juices of your stomach inside your stomach. We don't want that leaking out anywhere. We don't want that leaking out in between cells. So we want to make sure that there is a really tight junction between each one of those cells, and that there's absolutely no room for leakage, I guess, between the cells. So this is going to form an impermeable barrier. Those desmosomes are going to form a nice sort of flexible layer, give us some movement, kind of like your skin. We do not need quite that level of flexibility in our stomach. We want to make sure we have really, really tight junctions. So the next type of junction that I want to talk about is a gap junction. Now, the purpose of a gap junction is not to hold those cells together, but to allow for rapid communication between those cells. Gap junctions are going to be designed specifically for cellular communication. This is going to form when two identical plasma membrane channels join, and the channel of each of these cells is going to be lined by 6 plasma membrane proteins. Now, these gap junctions can provide strength to the cells, but really the point is to form these little channels that will allow small molecules and ions to pass through them. Now, this is going to be incredibly important when rapid, succinct communication is of the essence, specifically in the heart muscle and smooth muscle, where we need those ions to flow from one cell to the next to facilitate simultaneous contraction. We need the whole heart working in unison. We need very, very rapid communication, so that's where we're going to see these junctions, these gap junctions for cellular communication. So here we've got just a summary of those different junctions, adhesion junctions with those intercellular filaments between the cells. We've got desmosomes, which have the internal cytoplasmic plaques. They kind of work like buttons. And then we have gap junctions. Those are going to form an impermeable barrier. And we have gap junctions. Their primary responsibility is going to be plasma membrane channels. It's going to be for the rapid cell-to-cell communication. Another external feature that we're going to find, or a cell surface modification that we're going to find, specifically in plant cells, is that we are going to find a cell wall. So not only do plant cells have a plasma membrane to protect them, but they also have a porous cell wall that's going to be made up of cellulose. We're going to find that the cellulose wall is going to be the main component. It's going to give it strength and support. And what we're going to find is that the pectins in the cell wall allow the cell wall to stretch while the plant is growing, and non-cellulose polysaccharides to harden the wall when the cell reaches maturity. So in a plant, the cytoplasm is going to be connected by plasmodesmata. This is just going to be very numerous, narrow membrane-lined channels that pass through the cell wall. This is going to allow the cells to pass nutrients back and forth. So each one of those contains a strand of cytoplasm. It's going to allow for the transmission of nutrients in between cells directly. So it's these itty-bitty little channels that allow for the flow and the movement of cells from one channel to the next. Now, cells in woody plants have a secondary cell wall that contains lignin and even more cellulose fibers than that primary cell wall. That's the main difference between woody plant cells, or woody plants, and herbaceous plants is the presence of that second cellular wall that contains lignin. All right, and that is it for our chapter on cell membrane and structure and function. Please let me know if you have any questions, comments, or concerns. I'll be happy to go over anything in greater detail with you during our discussion time.